So as we go through and we look at DNA, RNA, and protein synthesis, you guys are going to see some hands-on activities that are going to hopefully clarify a lot of information that we're going through in the notes. So what you see here, if you guys have had siblings that have taken human bio before, they may have done the famous DNA poster project. Well, we can't give those parts out to you because everybody's online and computers and things like that. So I'm going to go through the parts and the pieces that we have here that you guys would be manipulating and talking to me about. I'm going to kind of talk to you about, and you guys will see some little assignments that go along with it. So in front of me, my poster project, I'm going to make things move on it. So you just have the piece of white paper just as my background. In the middle, I have my nucleus, and in that nucleus, I have a little section of DNA. So we're going to pretend that the rest is super tightly coiled. It's making up the chromosomes. There's 46 of them in each of our cells. So this particular DNA structure that we want to look at, we want to kind of break it down in terms of its parts before we go through and see how we can replicate it. Remember, DNA is deoxyribonucleic acid. It is found in your cells, the nucleus of your cells, and it's basically the blueprint that makes you who you are. Codes for all of your proteins, hence protein synthesis that we'll be seeing, as well as all of your traits. DNA is made up of nucleotides. So I can split this giant molecule down into individual nucleotides, and a nucleotide is one sugar, one phosphate, and one nitrogen base. In the middle here, the rungs of my ladder are my nitrogen bases. You have 4 different nitrogen bases in DNA. You've got adenine, thymine, guanine, and cytosine. And as we went through the scientists and notes of that, you guys should have seen Levine and Chargaff and talks about how these nitrogen base pairs come together and the bonds between them. So let's break it down a little farther. We just said a nucleotide is composed of a sugar, a phosphate, and a nitrogen base. You don't see sugars and phosphates here, but they make up the backbone of this ladder structure. So I have alternating sugar, phosphate, sugar, phosphate, sugar, phosphate going down both sides of my DNA structure. My sugar in DNA is deoxyribose. My sugar, phosphate, and nitrogen base makes up that nucleotide. If I have 4 different nitrogen bases, I have 4 different nucleotides. I have sugar, phosphate, thymine. I have sugar, phosphate, adenine, sugar, phosphate, cytosine, sugar, phosphate, guanine. The way that these nucleotides are strung together give you guys each your own individual genetic codes of A's, C's, T's, and G's. If we keep going down a little bit more, and I look at these particular nitrogen bases, we know that they are complementary base pairs and that adenine always pairs with thymine. I know that guanine always pairs with cytosine and vice versa, cytosine with guanine and thymine with adenine. If you are looking at them closely, one of the scientists that we talked about was Levine. And Levine sort of broke down the structures of our nitrogen bases, and he said 2 of these nitrogen bases, guanine and adenine, are larger base pairs. They are double ring structures. They are known as purines. 2 of them, thymine and cytosine, are known as pyrimidines. They are single ring structures, so you can actually see that they're smaller on this particular model that you guys have here. Whenever nitrogen base pairs come together, because we have 2 base pairs, complementary base pairs making up the rungs of our ladder, you will always join one purine with one pyrimidine. So one purine with one pyrimidine. And the ways that we know how they come together is kind of with Chargaff's rules. So Chargaff said, okay, well, within a cell, if I see adenine, I know that I have equal amounts. He discovered that there was equal amounts of thymine. So what that meant is that adenine and thymine were kind of bonded together. They are complementary base pairs to each other. My adenine is my purine. My thymine is my pyrimidine. They are bonded together with 2 hydrogen bonds. Every time I see cytosine, I have the same amount of guanine in there. They are complementary base pairs. One purine, guanine, one pyrimidine, which is my cytosine, and they are bonded with 3 hydrogen bonds between them. So remember, these hydrogen bonds are weak bonds. My nitrogen bases will always attach to my sugar or deoxyribose. So it's kind of like, here's my base pair, here's sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way up along both sides. Also within your notes, you guys should have remembered that we talked a little bit about 3' and 5' ends. So if I had given you these structures that look like this and I say, okay, without the numbers there, identify a 3' and 5' end. You can't do it. I didn't give you the makeup of your sugars. But I do know that this DNA strand runs anti-parallel. They run in opposite directions. So if I tell you one end is 3' end, what that means is that when we look at our sugar, if I give you the chemical makeup, and we started our oxygen, start counting carbons clockwise, my 3rd carbon is facing down towards the end. My 5th carbon is facing upward. This is a 3' end, 5' end. They're anti-parallel, they run opposite. If this is 5, the top of this strand has to be 3, the bottom of this strand has to be 5. And you're like, so who cares? Why is that so important? Because when this molecule goes through replication, we're making an identical copy of it. It will be read in one direction and the new strand will be created in a direction. So that's why these 3 and 5 primes, meaning the 3rd and 5th carbon in our 5 carbon sugar, play an important role. So let's look at replication. Why do we need it? Well, you guys are growing, you guys are developing, you guys are repairing, you need new cells. Those new cells need more DNA within them. So I need to make identical copies of it. So we're going to go through this process called semi-conservative replication. So this idea of semi-conservative replication goes along with your scientists Meselson and Stahl that we talked about. So we know that DNA is supercoiled and we need to make it into this ladder-like structure. There's a lot of enzymes and there's a lot more than what we're just going to talk about. The first enzyme that we have is going to take DNA from being this double helix supercoiled molecule. It's called topoisomerase. And topoisomerase uncoils it and makes it into this ladder-like structure. Now remember, this can't just happen at one spot, let's say the top of your chromosome, and work its way all the way down. This is going to happen at multiple spots along the chromosome, and eventually they're all going to connect. That way it's a lot faster of a process in order to go through this DNA replication. So topoisomerase put it into this ladder-like structure. Now what we need to do is we need helicase to come along and break our hydrogen bonds. So remember, there's 2 between adenine and thymine. There's 3 between cytosine and guanine. It's kind of like a little zipper. So helicase comes in, it starts splitting them open. And what it's doing, it's creating something called a replication fork. Replication fork is not a location because it's constantly moving. It's where those hydrogen bonds are being broken and my strand is opening up. So to start with, my replication fork might only be up to there, and then it opens up a little bit more, and it keeps going, keeps going, keeps going, keeps going, and there's my replication fork. My strand is open. Within your nucleus, you have free-floating nucleotides. Comes from the foods that you eat. You have sugars and phosphates and nitrogen bases all floating around in there. You have an enzyme known as DNA polymerase. DNA polymerase is going to read your old strands and call in the complementary or correct nucleotides to match up to that old strand. So we're saying the white is my old strand, helicase came in, created that replication fork that you see here. Now I'm going to have my DNA polymerase come in and start reading my DNA. DNA is read 3' to 5'. So this strand is going to get read in this direction towards my replication fork. So it's going to be created in that direction. I'll grab a little marker here for you guys so we can look at that just a little bit more. So like I just said, DNA is read 3' to 5' ends. So it is going from 3 to 5. It's going to be read in this direction, going towards that replication fork. And who's doing the reading is DNA polymerase. So DNA polymerase comes in and he is going to make new DNA. So DNA is made 5' ends to 3' ends. So it's made 5 to 3. So here it comes. Polymerase hooks on right here. He goes, oh, thymine, where's my adenine? Adenine comes in. They bond together with hydrogen bonds. Adenine with thymine, guanine with cytosine, cytosine with guanine. And polymerase, DNA polymerase is going up the chain, working towards that replication fork, and it's making my new DNA. So we said it's being read in this direction. It's being made in this direction. That new DNA strand is being made 5' to 3'. It's opposite of my strand that was just being read. Here I have a 3' end. This is my 5' end. This strand is my easy strand, I'm going to say, and I'll come back and tell you why in a moment. DNA polymerase is also going to read my other strand, right? So we're still going to make complementary base pairs. But look what happens here. Because I have to read from 3' to 5', I'm going to be working away from the replication fork. Now I have to get made away from the replication fork because my strand's being read in that direction. So let's pretend that my replication fork was only open to, let's say, right about here. That's where the replication fork was at that point. DNA polymerase comes in, he hooks up right here, he goes up, let's start putting it together. This would be my 3' end, the top of the strand. It's going to get made in this direction. So polymerase comes in, he hooks up, he goes up, there's a cytosine. Let's do a guanine. So he hooks in, there's a guanine, there's a guanine, there's a cytosine, there's an adenine, there's a thymine. He got made in this direction. DNA polymerase gets down to the end, and he looks back, and he goes, oh, my replication fork opened up some more. He's got to unattach from the strand, he's got to come back up and attach back on again, and start reading those base pairs again. To help him do that, because unhooking and hooking back on is a little bit of trouble, he needs to rely upon RNA primase. So it actually makes kind of this like little docking point for DNA polymerase. Don't worry about it, RNA primase is going to get kind of cut out and filled back in at the end. But what's going to happen is it needs to hook back on again, it needs to read it again, and it's going to make a little fragment. So those pieces come together, DNA gets read 3' to 5', it's going to get made 5 to 3. I got another little piece. So these are actually separate fragments at this time. My replication fork opens up some more. And it's got to come back in. Primase is there, my RNA, polymerase hooks on, it reads it again. So now it's going to make me another little fragment. If we're looking at this, the reason I said this strand over here was my easy strand. This strand over here is called my leading strand. It is following the replication fork. So when DNA polymerase comes on and hooks up to my thymine and into reading my old DNA 3' to 5', it's happy. It's going, okay, adenine, thymine, guanine, cytosine, boop, ba-doop, ba-doop, ba-doop. And he is following the replication fork, and he doesn't have to stop, break off from the strand, get back on again. He is called a leading strand. This side has to be read. It's still read the same direction, 3' to 5', but because these strands are opposite directions, it is getting read away from the replication fork. It is getting made away from the replication fork. Still read 3 to 5, still made 5 to 3. But polymerase isn't as happy here. He gets to the end. He has to break off, come back up because the replication fork opened up more, hook back on, go down, make another fragment. He looks back, the replication fork opened up more, hooks back on, goes down, makes a fragment. This side is called my lagging strand. It's not saying that, oh, left side's always leading, right side's always lagging. You have to see the way that replication fork is opening up and which strand is going to be continuous and which strand is being made away from the replication fork and making little fragments. So as polymerase came in and did all this, I now have these fragments, and those fragments are going to be glued together by what is known as DNA ligase. Ligase is kind of the glue. So by the time I'm done, I have two strands that are completely identical. Completely identical. TNA, TNA, A and T, G and C, my base pairs are the same. That is what replication is referring to. I'm making identical copies. We say that what you see in front of you is known as semi-conservative replication. Each strand has 1/2 old DNA, which is my white, and 1/2 new DNA, which is my blue. So at this point, I have twice the amount of DNA that I should have within my nucleus. So what has to happen to this cell? Well, this cell has to go through cell division. It needs to split. So I'll form a new nuclei. We'll go through the process of mitosis or meiosis. We'll talk about that a little bit more later on. And now I have two separate cells with the correct amount of genetic material in them. So this goes off. The whole cell itself is going to divide, and it will go off And now I'm just left with my DNA again. So now this is considered my original DNA, and we can go through and divide that. Depending upon the type of cell that you are, might happen every couple days, might take every 30 days, may not happen at all. Some cells don't actually go through and replicate their DNA. If this was a skin cell, your skin cells do this about every 28 days, unless of course they're repairing themselves. Constantly going through and making more. All of this process of DNA replication will take place in the nucleus. And the reason is because even though we say, oh, DNA is this really teeny tiny little molecule, it is, but it isn't. It's too big. It's double-stranded. It can't fit through the nuclear pores. And that's why our next step that we're going to see, we're going to talk about transcription and translation. The problem is, this is your code for everything about you. It makes all of your proteins, including your enzymes. But the idea of protein synthesis is going to take place out here in the cytoplasm. So I need to get this code, these A's and T's and C's and G's, out here to make your proteins. This is too big. We got to make a smaller version. And that's where we're going to come into transcription next time. So if you need to, go back, watch that again, write down your questions. We can chat about it more.